and welcome back to another A-level biology video with Starfish Revision. This video is going to be about lipids. It's going to cover the structure and function of triglycerides and phospholipids. We're going to look at the function of those two molecules in living organisms, and finally we're going to look at the test for lipids in food. Lipids are fats. They are found in foods like oil and butter. And as you can see, one of the most noticeable properties is that they don't mix with water. This is olive oil, and you can see it just doesn't want to mix with the water. The two main types of lipid we need to know about are triglycerides and phospholipids. Let's take a closer look at triglycerides first. Triglycerides consist of a glycerol molecule that has 3 fatty acid chains attached to it. Glycerol is an alcohol. Fatty acids are just long chains of carbon with hydrogens attached, with a carboxylic acid group at one end. Each fatty acid chain joins the glycerol by a condensation reaction. One molecule of water is lost for each fatty acid that is added. The bond between the fatty acid and the glycerol is called an ester bond. The fatty acid tails contain a lot of stored chemical energy. In fact, twice as much energy per gram as carbohydrates. This makes fats an incredibly good energy store. Phospholipids are the same as triglycerides, except one of the fatty acid chains has been replaced by a phosphate group. Phospholipids make up the membranes around and within our cells. Check out the video on cell membranes for more details. The key features of phospholipids is that their phosphate heads are hydrophilic, or water-loving, while their tails are hydrophobic, or water-hating, so they naturally form a bilayer, with the heads facing out and the tails facing inwards, and this creates a barrier between two areas. Let's take a closer look at fatty acid tails. They are long chains of carbons with hydrogens attached. If there are no double bonds, we say the fatty acid is saturated. This means we can't add any more hydrogens to the carbon chain. Carbon can only make 4 bonds. Saturated fatty acids stick together more strongly, and so lipids with mostly saturated fatty acids have a higher melting point. Unsaturated fatty acids have at least 1 double bond in the carbon chain. This double bond causes the chain to bend at that point, so the fatty acid chains don't lie together so closely, and so they have a lower melting point. It's true that fats in mammals tend to be more saturated. So we'll know that butter is solid at room temperature, as is bacon rind. These are the fats that are found in mammals. And that's okay, because at normal mammalian body temperature, which is about 37 degrees Celsius, these fats will be in a liquid form. It's only that when we cool them down to room temperature that they solidify. In contrast, plant fats tend to be more unsaturated, and that's important for them because they want their fats to be in a liquid form at a lower temperature, because the plant can't keep itself at 37 degrees. And the same thing goes for fish. So that's why people say eat a lot of fish or oily fish, because the fats in fish are unsaturated. This is because fish can't keep their body temperature at the same level as a mammal could. They don't want their fats to solidify in their bodies, so they really do have to be liquid at lower temperatures, and therefore they need to be unsaturated. Unsaturated fatty acids are more healthy for us to eat. If you'd like to know more about which fats are good for you and which fats aren't so good for you, I've left a link in the description below. It's outside of the A-level specification, but you can read it if you want to do a bit of extra reading on this topic. Finally, let's look at the test for lipids. This is called the emulsion test. First, add some ethanol to the sample and shake it vigorously. Then, slowly add water. If a milky emulsion forms, this indicates the presence of a lipid in the food sample. That's it for this video on lipids. I hope you found it helpful. Please like and subscribe if you did, and I'll see you in the next video. Thanks for watching.